introduce microorganisms that will actually stick around in this substrate, which gets me to the results that we arrived at. So firstly, we can look at these visuals of our tomato plants. In this first image, you can see that there's a lot of root growth. These first two images, really, there's a lot of surface level root growth, which tells us, again, that there's a drainage issue. These plants are investing more time growing their roots closer to the surface rather than attempting to grow roots further down into the substrate. However, as you can see in this image, some of the roots did penetrate all the way through. And as a matter of fact, at the end of this experiment, when we were cleaning up and removing the substrate from the system, we noticed that there were roots growing through the fabric mesh into the cavity, the LECA cavity below. And so it seems that some of our solutions did work at least partly regarding drainage. And here in the top right, you can see our tomato harvest. This is our this is initial, right before harvest. This is about 2 weeks before harvest. And the fruit looks amazing, as you can see, and appears very high quality. Now, in this slide, you can see a comparison between the count of green fruit and the count of red fruit at harvest after 6 weeks of growing in the system. So you can see here the non-inoculated perlite and inoculated perlite are not very different from one another. And these plants performed better in the green fruit department than the inoculated regolith and the non-inoculated regolith. So the microbes appear not to have had a dramatic effect, which tells us that either we were doing something wrong in terms of how we implemented the microbial inoculant, or perhaps these microorganisms simply weren't able to thrive in the lunar regolith simulant for whatever reason. But it's also indicated, this is also an indicator of stress. So you can see on the right here, we have our count of red fruit. This is pretty similar among all the groups. Whereas here, the green fruit is lower in the regolith treatment groups, which tells us that the next wave of fruit that would have been grown to maturity had we proceeded with a second harvest or a third harvest, we would have had less fruit from these future harvests. Whereas here we have a pretty similar count of red fruit, which tells us that although there does appear to be some stress, nevertheless, plants grown in regolith, as compared to plants grown in perlite, perform pretty comparably. And here we have an indicator of our fresh weight and dry weight of the plants, excluding the fruit. And you can see an absolutely noticeable trend between the perlite versus the regolith. The regolith treatment groups produced much lower foliage, lower amounts of foliage than the perlite treatment groups. Interestingly, as well, the non-inoculated perlite appears to have had, or excuse me, the inoculated perlite appears to have had a greater water weight than the non-inoculated perlite. And so this indicates that that the plants are producing more foliage or less foliage in regolith, but a similar quantity of fruit, which is again an indicator of mild stress, but not potentially ultimately detrimental stress, detrimental to productivity specifically. And lastly, I just want to touch on a follow-up experiment that has since been completed here at the SEAC with the support of the Spring Institute. Thank you, Patrick. So this experiment sought to test germination rates in lunar regolith simulant amended in different ways. So you can see these more brown lunar regolith simulant trays, this is mixed with cocoa coir, so coconut fibers. The more green trays are lunar regolith simulant mixed with spirulina. And the more white trays or gray, these are plain lunar regolith simulant unamended. Now, what's interesting about this is given the hydrophobic nature of the lunar regolith simulant that we discussed earlier, we expected sub-irrigation to be more optimal in terms of providing an even distribution and a thorough hydration of the soil. But what we're seeing here is there's a lot of inconsistency, even within treatment groups. So the normal unamended lunar regolith simulant sometimes is bone dry and sometimes is soaking wet. In this case, they all became soaking wet eventually, but the rate at which water percolated up through the soil was, or through the substrate, was different cell to cell. Furthermore, spirulina. If you use spirulina in a system like this, word of caution, don't. It is very hydrophobic. You're going to want to compost the spirulina first. You're going to want to process it in a specific way. Otherwise, you're going to end up with this anoxic sludge. It's nasty. It smells awful. And plants probably would not grow in it very healthily. And furthermore, the increased hydrophobic nature of this mixture renders these cells impossible to water, almost. Not totally impossible, as you can see, but very nearly. Alrighty, and that brings me to the end of my presentation.
Thanks a bunch, Adam. It's good to see that not all of your adventures were so maligned with the spirulina. No, it was really interesting actually to see the comparison between the perlite and the regolith growth treatments and how I mean the production of the fruit was not that different. That is interesting. I think we probably do not actually have time for questions, but the chat has been blowing up talking about the merits of regolith and soil-based agriculture versus hydroponics. And so maybe since you were somewhere in between, you can add to that conversation. Yeah, thanks a bunch for your presentation. I really appreciate it.
Absolutely.
The next person on the list is Ted Tagami from Magnitude IO, and he's going to be telling us some of the lessons that he's learned from 10 or more astrobotany experiments to the ISS. Hey, Ted.
Hey guys. Oh my gosh, my brain is full. Here, let me share the screen. I'm gonna have to watch these recordings multiple times. My
Yeah.
background's art, it's not science or engineering, as you'll soon discover by the time I get to the end of my presentation. But yeah, we get in trouble here. Hopefully you see that. I'll make it large.
Yeah, we can see it.
Great. All right. So we're powered by curiosity. That's pretty much how it started. A lot of things going on. We're an education company and we put things in orbit. And actually, I'm going to have a little activity for you guys before your coffee break. Whether you made your coffee the old-fashioned way, you grow it hydroponically or in lunar regolith. I'm sure you're looking forward to that break. This is what we do. We connect students around the world, science centers, libraries, and museums to authentic research in space. Our fidelity has been really, I would say, low. My whole impression when I figured out we could do is like, let's just get started. After 10 missions, we've gotten a little better, and I'm really grateful of the response we've gotten from research. Talk a little bit more toward the end, and really getting to the omics side of things and really what's happening. The way I look at space right now, it's in crop production or plants. It's either looking at those wonders, the mysteries, and trying to uncover what plants do and all those chemical reactions and the proteins and what have you, how those stressors might be affected under these different environments. And the other side is like, let's just grow some food, right? And so that macro and micro, we're definitely a little more on the macro side with our students. I think really, I like to say K to gray, but with the assistance and support, I do have an ask here at the end. It's pretty important for us to kind of move forward. This is what we do. This is the ExoLab. It's an analog experiment, the same size as our original mission that went up to space in 2017. It's just 20 centimeters tall by 10 by 10. And we've got them all over the world, anywhere from Australia to Zambia, and they're measuring just ambient conditions. So they're not very sophisticated. Carbon dioxide, temperature, relative humidity, light, commercial off-the-shelf LED array here, and a camera that's capturing images. So we average that data every minute. We send it up to the cloud. We capture the images once an hour, also goes to the cloud. We then have a time series data of that particular lab. We can compare them against all the labs, and probably most importantly, the mother experiment that we send up to space. For the first time this year, we're actually going to be adding machine learning to all this. So maybe figure out a little bit more about optimal conditions terrestrially, and perhaps seeing something that's happening in space as well. So I'm going to give you guys a little bit of an interactive here. We can use chat for that. This is normally a breakout session for like year 9 and year 10 high school students at like symposiums and conferences and stuff like that. We're going to do it in 1 minute though, because I just really want to kind of give you a sense of where we're going when we think about ecology and thinking about some of the challenges we face. The way I'm looking at it right now, I remember them going to the moon in 1969. I was just 6 years old. These kids that are graduating from high school today, when they're my age right now, the year is going to be 2068. What's going to be happening in the world when they're my age, right? And so we've got a long path. I'm trying to think not incrementally, but really our namesake, thinking in terms of orders of magnitude, whether we're reducing cost, increasing reach, or maybe thinking about things orthogonally to the way people are normally looking at it. So here's our 1-hour design sprint in just a minute, okay? And we basically will be using a design concept, whether it's Stanford D School or the Royal College of Art there in London. This is some iSTEM work from New South Wales Department of Ed. So here it is. You got 30 seconds. Put in chat. You're a crew member on a starship. Space travel is slow, and you're going to be stuck with your crewmates for a long time. Let's say we're on the way to Mars. What critical factors must be considered? Go ahead and light up chat. Put some stuff in there. I'll give it a little count for roughly 30 seconds. And just kind of see what's on top of mind for everyone. I imagine it's going to be plants and what have you, but there's probably some other things as well you're thinking about. Right. And let's see how many of you are actually here and present. I guess it's another way for me to check. Roll call. Great. I'm watching pop up here. That's good. Give me a chance to also compose myself. I'm pretty excited to be presenting all you guys. I'm probably the most naive guy in the whole 2 days. Exactly, right. Netflix and chill, right? I mean, there's like how many movies can you watch on the way to Mars? So the reveal is, let's see if I've got these in the right order. I might have a more of a dystopic view here, but I think I've got the reveal here next, coming up next. Yes, there's your spaceship, right? Everything our students were thinking about on that voyage, you usually give them 3 minutes as we get into that 1-hour session. I want them to realize we are on a spacecraft right now. And there's a lot of things we need to think about, and the things like boredom, your investment portfolio, food, clean air, clean water, companionship, all those things super important. But here's something that's really important as we look at this is, again, I'm not a researcher, right? Not a scientist or an engineer. I'm just kind of more of an art guy that likes to cause the good kind of trouble. When we think about this next picture here, I want you to, instead of looking at what you see, imagine it as plants. Is that what we're doing? Is that the future? That's a Chilean zoo, and that animal there, Taco, was in that prison, if you will, for 16 years. It's alive, it's living, or is it, right? What are some conditions that we need to think about when we think about even something as lowly as the plant? Because they're not really lowly, right? They're pretty amazing. And they've got all those other things, the mycorrhizal fungi, our living soil. I mean, the other nearby neighbors don't have anything like this, right? So I think we really have to think about everything in context. Anyway, let's get past that part, but I want you to remember it when you're doing your work. Here are some of the challenges for the students as we go through that 1 hour. What's life support like? What's radiation? How are we going to handle that? Microgravity or partial gravities? What are some challenges? And I'm doing something called Crazy Eights, where they're given an 8x8 piece of paper or a deck of cards, and they're filling in, looking at either any one of these challenges or all of the challenges, and giving themselves just 1 minute to quickly write what they think in terms of their mind. Psychology, how your well-being. How important is that? Food production, of course, everything that I think most of us are talking about and interested in here today. Healthcare, waste management. I think NASA just made an announcement, some funding looking at trying to solve some waste management issues. The fact that we fill up an entire cargo vehicle to burn up in the upper atmosphere every few months, is that really the right way to go about it? Probably not. That's what we got today. But again, by mid-century, what's it supposed to look like, right? In situ resource utilization. So that's really quick 1 hour in 1 minute. Sustainable development goals helps us focus. So whether we're talking to students in Senegal or in Sri Lanka, whether in Tokyo or New York, the idea here is this as a lens really helps us focus, whether it's zero hunger, quality education or equity, life on land or underwater, really any of these, we look through that. They might be side quests. If we think about this like a video game, we have that main quest line. We're putting this plan in space. But these side quests really, I think, invigorate thinking and help students align and for us to meet them where they are. So it's very important for us to think about the SDGs. And we've worked with the UN in the past with something called Opportunities for Youth in Africa, a big challenge there. Get them excited. And when you think about space, it's like, well, how practical is that for them on Earth when they're thinking about food and food security? So here we are. I like to say that this is a seed program where science and engineering education is through design. That's kind of really more where I'm coming from. And the rest of this deck is pretty much going to be our origin story real quick. Exolab Project Cadence. Exolab is that product that connects to the space station. Some upcoming missions. We got 2 missions launching on SpaceX 31. Supposed to be the end of the month, but that Milton hurricane, hopefully everyone's safe that was down there in Florida, put a little delays in the launch. Don't have an actual date from NASA. And then a request for cooperation because I don't know a lot and you guys do. How might we extend our learning for that next generation? So the origin story real quick here. My buddy Tony came to me just after my 50th birthday and says, we can sell advertising in space. And I'm like, yeah, what? No. But within 4 months, we formed Magnitude with the idea of focusing on education. My kids were in high school. Tony's children were in middle school, and it just felt right. The next generation science standards were just introduced in the state of California. And within, I guess within a year, we had a project with a company called ArduSat, which is now called Spire, publicly traded satellite company. But that CubeSat didn't work. It actually had 3 launches that failed. And we had several hundred schools that we had to refund. Oh, wow. Talk about disappointing. Talk about just fold the tent and go home. We've only been doing it for a year. But in that discussion and meeting all the amazing people that are involved in space, we came across Twyman Clements over at Space Tango and the idea of Tango Labs. And what we could do is basically it's the same form factors, but we had a chance to put something in space. Intermodal transportation, right? Instead of a 40-foot container on the back of a train or back of a truck or a ship on the ocean, fill one of these up. You can send it anywhere in the world, and everyone from point A to point Z knows what to do with it. And intermodal transportation for space, Nanoracks has them, Tango has them. There are probably other folks. They're basically looking at the mid-deck locker and how much can you squeeze in one of those. So we've taken a vertical farm, squished it into a shoebox, and we put it in orbit. And talk about problems, talk about challenges, right? But hey, they're doing it, right? Eden is down there at the South Pole, and they've got it working. But how do we make something even smaller? How do we crush it down? So here we were, October 14th. We did that announcement there. It's no longer around. Berkeley Biolabs, Dr. Ron Shigeta gave me infinite wisdom. He said, because I wanted to look at entomology, I wanted to look at microfluidics, I want to do all this stuff. He says, focus. So he holds up an Arabidopsis and he says, focus on this to start. That's what our first lab looked like, launched in 2017 aboard SpaceX 10. And guys, I should have put pumpkin seeds in there because we roasted those seeds in orbit. It was pretty bad, pretty sad. But we kept on going, right? We actually launched something. Cadence, surprisingly, by the end of that year, we're able to launch again. And then we've pretty much been launching, I'd say average about every 18 months. We're going to have to take a pregnant pause next year because it's actually getting more expensive, an 8-fold increase in our costs. Not because space is getting more expensive. It's actually cheaper, as you guys know. It's the payload resources. Getting to the space station, there's less places to be put. And getting your download, you're getting your payload to return home is also a difficult challenge. We were bumped. We were supposed to fly in March. We wanted cold stowage return. sorry, there's something else coming down that was more important, so we had to wait. One time we got bounced because the mice needed food. So, that's what happens when you're in education, right? Here's just kind of our mission heritage from SpaceX 10 in 2017, all the way through Sativa and Truncatula here at the back end of 2022. The cadence has been regular. The exploration has been all over the map. We started with Arabidopsis. We went to some microgreens, and in 2019, a farm boy turned educator in New York said, "Hey, do you know much about the symbiotic relationship between bacteria and your legumes?" I said, "No, tell me more." And we've been studying that. I wouldn't call it success so much as that we're getting these in orbit. But I'm really thankful that Purdue and Professor Porterfield's here can share a little bit more about. We're going to turn over our flight specimens to him this year to really kind of get some omics out of this. But that's just some flight heritage. Of course, you get up on a Northrop Grumman mission, you come home on a SpaceX Dragon. We were really, really lucky. That was not planned at all. We thought we were just going to go back on a regular flight on our 2021 mission on Northrop Grumman 15. We came on a Crew Dragon 1, and that was just like, wow, part of history. Here's ExoLab 11. This is one of the 2 missions that's going up here, probably early November. SpaceX 31, we're growing the model organism for your pulses, for your legumes. It's got a simple diploid, I guess, genome structure. It's already pretty well mapped. We've got a seed line from the Australian Pastures Gene Bank, Jamalong A17. So it has a good heritage. And we're just going to see what happens in flight. We're going to have 30 days nominal activity in orbit, and these are all autonomous. Once we dock with the space station, within usually 6 hours or so, an astronaut assigned to us will float over and grab the flight bag, go over to that mid-deck locker, open up the locker door, pull out a big data card, put our lab on it, slide it back in, boot us up, and then kind of float away after we make sure we've got telemetry, our environmental data, our imaging data is coming down. And then usually within 48 hours or so, we ask our schools and science centers, our libraries and museums around the world to go ahead and start their experiments. And now, in conjunction with that experiment, they're seeing in real time what their labs are doing on Earth and what's happening in orbit. And then we connect them with a weekly session. And there are a couple folks that are on the in these sessions here that have joined us gratefully to share their expertise and understanding about the wonder of space exploration, and all the various careers that can happen here. We'll ascend on this case in 4C, come home in -80. We did some fixes on the last mission, and we had some pumping issues, and it kind of started frying some electronics, and it was pretty sad. So we went for simple. That on the right-hand side, 2 equivalent is the ground trial after 30 days of growth of the Truncatula. You'll see a big agar block there. This is what it looks like. I think we've got pretty good root development because we were concerned about the drowning or suffocation of those roots. We've added that arsenite there as a punch out to create some breathing space for the plant. And something you don't see here, but there, these 2 tubes here, we're actually pulling out when that gets a very tiny chamber. When that moisture really builds up, I thought, wouldn't it be great, guys? It's essentially every mission's changed since the very first one. I would love us to kind of find some way to reduce our humidity. So we're running a desiccant column here. When the humidity gets a little too high, we'll flip that on. We'll pull that wet air through a desiccant tube. We'll dry it back out, put it back in the chamber. TRL on this is probably a 2, maybe a 3. And it's doing better than it should. And the volume probably needs to be bigger, but we're going to set it right now. We're ready to fly. It's actually starting to pull the moisture out of the agar block. So we've tried to find ways to minimize the surface exposure of the agar medium. Again, set and forget. So there's no pumping, nothing like that at all. Powerhouse 1 is another mission also flying. I just thought I'd mention it. This is a 3-year project with the Powerhouse Museum, a brand new 30,000 square meter museum equivalent in the capital expense as the Sydney Opera House. It's out in Western Sydney in the Parramatta region, the new geo-center of that metropolis. You've got the new airport that's going in out there, and intermodal rail lines going along the west side of the city, pretty far out. So this is an inaugural kind of event for them as they open their doors next year. Just happens to coincide with the International Astronautical Congress that convenes in Sydney in September. You can't tell my background's more in brand engagement and marketing. This all aligned up just absolutely perfectly. The students came across wanting to study algae, and so that's what we're doing here. And working with Dr. Harvey Bates from from the university there at Sydney, University of Technology, Sydney. We're using a derivative of his phenobottle, basically measuring the chlorophyll A through a photonic response using a fluorometer. And we're not using a cuvette or anything like that. There's no stirring. It's going to be kind of funky, honestly. And I'm curious to see what those algae are going to do. Are they going to clump together if they're under stress, or what's going to happen? Normally, they would just settle under gravity. It'll be interesting to see that. The students are exploring in a broad sense what might happen here. And here's my ask, everyone. Cooperative fundamental research. And so when I think about getting down to the younger ages here, zeros and ones is all I'm really interested in. It's very expensive for me to fly missions to space, and I'm not really doing the best job from a research perspective. But instead of being the tail wagging the dog, if we can think about cooperation and getting data that could be more public, and I know how important it is to publish your papers. I mean, that's gold for you guys, right? I'm not an academic, but if there's a way we can open this up and we can think about common missions that anyone in the world could be joining along in. And maybe the perfect one is when we think about heading to the moon and talk about the inspiration and the potential, the imaginations that are going to get fired up. Whether or not there's actually any meaningful science or engineering, it's that imagination power that I think is very real for us. But obvious benefits, right? Cost reductions and faster research. I mean, we can iterate more rapidly. And there's that sharing, that culture of sharing and both the successes and the failures. Like 2 days of what we had here, but imagine that over multiple years. And of course, using the TOPS as an identity there, open science data repository, whatever we can do, every work that we're going to do off of ExoLab 11 is going into as an open data science. The work that Purdue is going to do for us on the omics side will be there as well. Any of the imaging data that we're going to put out there for machine learning will also be available. Just our partners, have to be thankful for the National Laboratory helping us do it. Tango, and of course, these other folks here. I'll just end with this, the Goldilocks equation, right? Too hot, too cold, just right. Patrick, I see you popped in. I'm gonna turn it over to you. Thank you, guys.
No, thank you, Ted. That was fantastic. I love hearing from other parts of the industry. This is the cross-disciplinary stuff we're always talking about. So thanks for the work you're doing for education and bringing space to people. I know we've had a chat in the past about the Exoterra and the possibilities for it. I really love this experiment. We do have time for a question or two if anyone has them. I see you have a couple comments at least in the webinar chat. I think Joya has some tips for you maybe. I guess if I'll throw you an easy question because we're coming up on the coffee break here. I'm curious if you've ever flown, like what do you want to fly in ExoLab, and have you done that yet?
Have not yet. I'd like to fly myself. I can't fit in next to
All right.
But as far as plants, I guess I'm looking what everyone else is doing. And I'm enamored right now by the leaf project. We've got
Mm-hmm.
Arabidopsis, Brassica rapa, and a curious plant, Wolffia, right? Duckweed.
Exactly.
And something as we talk about strawberries and what have you, and I've been talking to really smart folks like you guys. And one of the challenges I think is usable mass. If you've got a substrate that you're using, can you use it all? Can you reuse it? Like
Mm,
the pillows right now, the pond system, you're throwing a good portion of the mass away. Can't use it again. Infection. If you look at growing strawberries, those vines, those leaves, the roots, really can't use them again. But that fruit is so delicious. So someone described it to me as a chassis. They look at the organism as a chassis. How might they genetically modify that to produce what they want, whether it's pharmaceuticals, whether it's materials for construction, whether it's food. And I'm really excited about that. And so I'm kind of leaning toward what I'm watching the real smart people do and looking at those plants. They chose them for a reason. They're going to the moon on the Leaf Project. So Wolfie, I think, would be something I'd love to do. Just throwing it out there for you guys, since I'm not flying anything next year, my open challenge to the students around the world is, what would you like to fly and why? And it can be the silliest of ideas. And of course, we're going to be focused on plants or fungi or what have you. But the idea here is to start that conversation and then hopefully weigh in from experts like yourselves. We can really think about what we should be thinking about when we go off world. We're going to need to bring a lot more about Earth with us, including some of that living soil, I think, to really make it worthwhile. Anyway, Patrick,
mhm.
thank you so much for letting me speak.
Yeah, of course. Thank you, Ted. So here we are on our last coffee break. It's going to be, I guess, 13 minutes now. We're going to reconvene at about 2:25 Pacific time. And then we have another hour and a half of talks, and we're finished with this edition of the workshop. So grab that coffee, re-energize yourselves, and I'll see you all back here in a few minutes. Okay, it's time for the last afternoon presentations. We've got a talk queued up from Adriana Sanchez about mushroom waste digestion for space applications. Hey, Adriana, welcome.
Hello, how are you?
Doing great. I'm glad you can make it.
I'm going to share my screen. There we go. All right. Okay. I guess we'll just do it this way. That's okay. That's fine. Hello, everyone. I hope you're all enjoying the Space Ecology Workshop as much as I am. I'm honored to have been selected to present at this year's event. As Patrick already said, I'm Adriana, and I'm an undergraduate student at Purdue University. I work in Dr. Porterfield's lab as a microengineer, and I have a passion for bioregenerative life support systems and using biological systems as a way to provide solutions for waste management. I'm also the hydroponics lead for our Astro USA club, our search club at Purdue, where I'm working on building autonomous hydroponics towers for our first student-led, fully closed-loop, self-sustaining architecture. Today I will be focusing on microremediation and explore how we can maximize its efficiency for sustainable waste management in space environments. As we embark on the next era of human spaceflight, understanding how to manage waste is critical, and knowing how to deal with this waste effectively will ensure the safety of our astronauts. Okay. Mushrooms have been breaking down waste for over 800 million years, and their remarkable ability to develop specialized enzymes to break down their environment is unparalleled by any other biological kingdom. The great decomposers of our planet continue to surprise us every day with their adaptability. The discovery of Paragenodontium album at the Great Pacific Garbage Patch, where they thrive on plastic pollution, or Cladosporium sphaerosporum, growing on the walls of the nuclear reactor in Chernobyl, are prime examples of their resilience. The key takeaway is that mushrooms always find a way, not only to survive, but to thrive in harsh and seemingly desolate environments. They don't need advancements in human science to learn to break down all the toxins that we introduce into the environment. They don't even need the safety and security of a stable environment to keep their phylum alive, and some fungi have even been able to grow in anaerobic conditions and developed hypoxia tolerance. All they need is a carbon source, and as soon as that mycelium come in contact with a potential source of carbon, it immediately gets to work breaking down waste. Anywhere we go, we're going to generate waste. Whether it be organic or hazardous waste, it is a part of life. With this in mind, we must consider the waste generated during space missions. In LEO or low Earth orbit, supplies are delivered to the ISS and produce a significant amount of plastic, food packaging, clothing, and many other miscellaneous wastes. The sustainability and resource management in space is a huge hurdle for deep space travel, as well as inhibiting Mars and other rocky celestial bodies. This presentation is meant to serve as an encouragement to you all to implement microremediation techniques as a main source of waste management. Okay. There's a sort of waste management hierarchy that I want to highlight today. First and foremost, we must prioritize waste prevention. So reducing consumption and minimizing any waste generated until we're left with basically the bare necessities, which we kind of already do, but there's still some things that are not reusable. So the next is to reuse and recycle materials. And though this is more challenging in space, it's still important. The reality is that we must operate at maximum efficiency most of the time, and this makes it really difficult to reuse components that may only be effective for a limited number of uses before they begin to lose efficiency and need to be replaced. Another point I want to bring up is that although something may be reusable, the risk that it imposes on the crew may outweigh the need to recycle it. So things like medical supplies. The fourth step in this hierarchy is recovery, which involves converting waste into energy or repurposing it to create new materials. And lastly, there's disposal, which we want to avoid, but disposal is less than ideal considering how mushrooms can assist with all types of carbon-based waste and can drastically lower the amount that gets tossed. Before we are able to establish growth in space or on another planet, we will need to send prepackaged food, which requires careful storage. Currently, food packaging consists of a 5-layer co-extrusion consisting of nylon, ethylene vinyl, tri-layer of polyethylene, and linear low-density polyethylene. Additionally, like I said previously, we must send medical kits, which are full of many different kinds of plastics, things like syringes, gloves, nylon sutures, catheters, medical waste bags, and many other medical devices are dangerous for us to reuse. They're made out of plastics like polypropylene, polyethylene, polyvinyl chloride, and polyamides. There will also be plastic waste generated from feminine hygiene products, and all of these materials represent potential carbon sources for fungal growth, all of which are discarded after one use. So there's something that can be done about these kinds of waste rather than just jettisoning them off into space. I would also like to emphasize to you all the urgency of developing effective and sustainable waste management strategies for future space missions. Not only can it help with our goals of inhabiting other planets, but it can create a cyclical economy here on Earth and make greener living more accessible. Okay. So when looking at the different applications for mushrooms have for waste management in general, I was interested in using mushrooms to reduce textile waste. And here we have a picture of oyster, blue oyster. And oyster is it's been really good at degrading many different kinds of waste, like plastics, which is surprising because it's an edible mushroom. It's not something that's used for waste disposal. And here I have some pictures of it growing on a bag full of cotton scrap textiles. And you can see the fruiting bodies as well as the mycelium on the inside. And it's completely covered this entire bag. Even though it's not an ideal substrate for this mushroom, it's still working pretty effectively. A promising candidate for mycoremediation, especially with plastics, is Pestilopidiosis microspora. It is a unicellular heterotroph that, like all fungus, it releases an enzyme into the environment and it breaks down the molecules, which it then absorbs as nutrients. This fungus reproduces by releasing spores, and it was discovered in 1880, but its polyurethane degradation was not discovered until 2010. And since then, researchers have found that this fungus can digest polyester, polyurethane, and polyethylene, making it an ideal candidate for addressing plastic waste in space. This mushroom can also digest multiple types of plastic simultaneously. So there are some mushrooms that can digest plastic as well, but they need to be isolated to one type or they won't fruit at all. In my research, I've been focusing on optimizing a strain of the chosen fungus. The growth method I've chosen is to suspend Pestilopidiosis microspora in a liquid medium that uses different types of plastic as its carbon source. To test the effectiveness of this fungus, I've chosen to use spectrophotometer to analyze the degradation of the different plastic materials over time, and this allows us to measure change in absorbance and quantify the extent of the plastic breakdown. So let's look at some more pictures. Here we have our liquid substrate with syringes, and then on the right here, I have another with acrylic yarn. This was just extra media that I had, and I decided to test it out anyways. This one was not like an official experiment. And here we have pictures of the mycelial growth inside of the said chambers. And here again, we have with the acrylic yarn, the fungus growing on top. And this one, because it's in this liquid media, we haven't really seen much fruiting bodies. It does release spores, but it doesn't necessarily have fruiting bodies. It's a lot different than the oyster mushrooms. Ultimately, this research contributes to the development of sustainable waste management strategies in space by addressing the critical challenges that we face in creating biogenerative life support systems. Our primary challenge now is to move beyond the confines of low Earth orbit and work towards establishing a continuous lunar outpost and sustained human presence on the moon. In the future, I would like to test the edibility of these mushrooms after they've degraded different sorts of plastics that have different toxins in them. as well as turning them into more useful materials. So things like clothing or things that can be used around the habitat to fix components, if that's even possible. I know that for sure you'll need to have a good HVAC and air filtration system, and a lot of those use carbon filters, and these could be used as a replacement for those carbon filters. Mushrooms can but won't always leach toxins for their environment, and it depends on the colony and the strain of the mushroom. So if we can take mushrooms that have been growing on plastics and don't absorb the toxins and are still edible, and we keep reproducing those, we can create more efficient mushrooms at degrading toxins that won't release them back into the environment. And then here again, we tried with polyester scraps. Well, one bag with millet and casting mix, as well as another one with just millet. And surprisingly, the bag that only had the millet had more mycelial growth. We didn't really see any on the one with the casting mix, but then again, these experiments need to be repeated multiple times, and mushrooms are very finicky. So thank you guys all very much.
Okay, thank you, Adriana. That was a really excellent presentation. I liked your the theme of your slides actually. That was a pretty nice Mars background. Okay. While we're waiting for people to, I see you got a lot of emojis, which is really nice feedback. Remind in there. If we've got some questions from the audience, I'd love to hear them. I guess we got one from Morgan already, I think. Have you explored growing these fungi in a mixed community or only in isolation? And then how do you feel about the possibility of other organisms aiding in the degradation process of the mushrooms?
So far, we've only grown them in isolation. With mushrooms, if you have other mushrooms present in the same like substrate, they will compete with each other. And so to make it more efficient, it's better to have a single mushroom by itself, as well as like other organisms. I haven't looked into it, but there's probably some organisms that grow pretty well alongside mushrooms. The danger with that, of course, is that there can be contamination. All of this stuff has to be autoclaved first before we start growing it, or there can be there's a risk of mold, and then the mushrooms won't take hold at all. And so it's really important to keep things very clean when we're working with mushrooms. But yeah, I'll have to look into organisms that live alongside mushrooms in the forest already.
Yeah. I've got a question. I saw that you were doing some experimentation with strange substrates. And in addition to the normal ones like millet, for example, what do you think is going to be the ideal substrate when you're living on a space station, for example, and you don't have access to the typical substrates like wood chips or whatever?
I think it's less about what the ideal substrate is and what's more about the ideal mushroom type that we're using. So if we can find, like I said, uh, making the strain stronger, making it better at digesting these plastics faster. That way, as soon as it comes in contact with whatever the plastic is, maybe we won't even need the liquid culture. We can just put the mushroom right onto the plastic and it'll start degrading. So I think it's more about picking a good mushroom rather than the ideal substrate, because really we want to use these to manage waste. Uh, Outside of plastics, we also want to focus on like using mushrooms to degrade human solid waste. There's a lot of mushrooms here on Earth that grow primarily in things like cow dung. So
Mhm.
if that can be used in space as a way to degrade human waste and then be turned into maybe something like a nutrient solution for hydroponics, maybe that's a way that we can close the nutrient loop.
Sure. Yeah, that's good thinking. Joya had a question specifically about those fungi that are eating plastics. How complete is that degradation, or do you have to use like multiple rounds or multiple species to completely consume the substrate?
So from what I've seen, it doesn't completely degrade it. It kind of starts the process, but there are some like the Pestaloptiosis in other instances, not me specifically, where I've seen it completely degrade the plastics, but it's still inside of the liquid media that they're using, so you can see the components and the enzymes that the mushrooms release to break down the plastic.
Okay. Oh wait, how are we doing on time? Oh, okay, we still have 5 minutes. Yeah.
Yeah, I see them.
Uh, oh, that's kind of interesting. One of the suggestions in here, well, it's, I guess maybe if we were to put this in question form, do you think if microplastics were suspended in a water solution, that the mushrooms would still be able to get at that and degrade them?
Yeah. So I
Yeah.
think that the mushroom that I mentioned earlier, the one at the Great Pacific Garbage Patch, it actually it lives in the water, and it first starts out with the smaller pieces first. It doesn't necessarily pick the pieces, but it's very much like it's easy to break down the smaller pieces than it is the big pieces because it has to get through that outside layer of the plastic first before it can really release its enzymes, and then it'll start to absorb the solution that it makes.
Mm,
So microplastics is a perfect example of what we could use as nutrient solution for these mushrooms. The processes that I have been doing, I think that I'm not using enough mushrooms in terms of how much plastic is inside the solution. Um, I think that to do complete degradation, we would need a lot more mushroom mass compared to the
sure.
amount of plastic that we're using.
Okay. Do I guess if you're working on mushrooms, you probably like eating them, I'm assuming. Is that a good assumption?
I personally do not like mushrooms,
Oh no.
but I will eat
Um,
them. They are good for you, so.
okay, so it's in the lima bean category. Uh, Um, I was bringing it up because I've recently got into mushroom foraging, and I live in the Pacific Northwest, and so it's the perfect time of year for that. And you can just find dozens of species of edible mushrooms just out walking around. I wanted to ask you about like what other kinds of mushrooms other than blue oysters are you really interested in cultivating and why?
So we use turkey tail because of its health benefits as well as lion's mane and reishi. Reishi is a super like a hardy mushroom. Like it doesn't matter what substrate we've used so far, it can eat it and it stays living and it fights these other organisms off a lot better than the other mushrooms that I mentioned.
Wow.
Like the Pestilopiosis microspora is very susceptible to getting contaminated as well as lion's mane. A lot of the white fungi are kind of susceptible to that. But
Hmm.
yeah, reishi is a big contender because we found that it has more melanin in it sometimes, and we've seen with the mushrooms at Chernobyl that the more melanin that is inside the mushroom, the better it is at protecting itself from radiation. So hopefully
Hmm.
maybe we can use it to grow clothes in the future for spacesuits and things like that to help protect
Oh,
against radiation.
yeah, interesting. That could be cool. I do like micromaterials. I think here's a question from Jane. It says one side is degrading the materials that we have, but the other side is selecting and designing materials for degradation. Have you thought about what an optimal substrate might be? I know you were saying choose the mushroom, but like if you had to.
Yeah. Um, Again, it's still kind of the same thing for me. It's just which mushrooms digest the which substrate the best, but I haven't thought about designing materials for degradation more because I think that collecting the materials that have been produced as waste already is kind of the main focus for me. So
Mm-hmm.
things like textile waste or plastic waste or human waste, anything like that would be is how can I find a way to break this down so that it can be reused and it won't destroy ecosystems anymore.
Yeah. Okay. I think that's all the questions for now. Thank you so much for this presentation, Adriana. It was excellent. Um, we're going to move on to Kevin Schaffman, who is going to be telling us about phosphorus and potassium solubilizing endophytes as an approach to in situ resource utilization. Hey, Kevin.
Hey, can you hear me?
Yep,
Cool. And I'm hoping you can see my presentation. Oops.
indeed. Yes.
Cool. Hi, so I'm Kevin Chapman. I'm a PhD student at the University of Washington, and my research is how endophytes, which hopefully you know what those are now from Morgan and Dr. Doty's talk, are bacteria that live within plants, and I study how they dissolve minerals and make them bioavailable to plants. I'm currently trying to figure out how to apply my research, which has mostly focused on sort of earth agriculture to non-earth agriculture. And because I'm a relatively young grad student and also I'm now just starting to approach this subject, I don't have any data to share, but I'm going to talk about like the feasibility and the idea behind this. And I'd love to hear you guys' feedback on any of this. And I'm super open to collaborations, and my email will be at the end. I also want to mention, while I will be talking primarily about Mars, most of what I will say is also applied to the moon, and I will indicate when when the moon is different. So think about, we're here on Earth. We're trying to think about how we need to live on Mars, or what we need. And we need shelter. We need somewhere to live. We need a way to run that shelter. We need food to feed our astronauts. We need a closed loop recycling system to make sure we don't poison our environment, to utilize our resources. But if we think on like a basic level, what we need is the elements of life. And so if you remember back to like high school biology, you may have heard the acronym SHNOPS. And so these are the nutrients of life. However, the micronutrients are also quite important for, you know, balancing transporters or enzymatic function. However, we generally don't think of these as the elements of life, but they're equally important. And so if we're talking about in situ resource utilization, which I think we all now understand why that's important, what would that look like, and how could plants fit into this? Well, in this scheme here, I have plants basically uptaking nutrients from regolith, which can then be converted into that circular recycling system. And so, you know, we often talk about how we can, you know, bring nutrients from Earth, and I'm sure that would work for early colonization efforts or early bases and stuff like that. But as those colonies grow, that's going to become more and more difficult. We really will need to start relying on the nutrients that is available on Mars. And so let's focus in on this idea of transferring abiotic nutrients to biotic nutrients, or this in situ nutrient utilization. And on Earth, we do this, or I guess plants do this. We just call it by a different name. This is basically primary ecological succession, where we have these bare rocks turning into these developed ecosystems with plants and fungi and bacteria and all that stuff. So let's think about an example of this. My favorite place to kind of talk about when it comes to ecological succession is Hawaii, because you've had all these lava flows, so you actually get these different ecological successional stages because every so often lava comes in and resets it. And so this is a place that's really early in ecological succession. I took this picture this summer, and it's 6 years after a lava flow. This is on the Big Island. It's on the Puna coast, if you know where that is. And it looks really desolate. So, I mean, I don't know, if I change the sky to red, you might think it's Mars. But it actually isn't desolate. If you look closely, there's plants absolutely everywhere. And this is only after 6 years, which I think is quite impressive that these plants are able to establish this quickly. You can even start seeing the earliest plants after 2 years. So then if we fast forward 60 years, we will start to see like the environment progressing a little farther. Now we have like a little bit of grass going on. We have some relatively large trees. And then if we let this run for another couple hundred years, we'll get a developed forest. And so how does this happen, right? Let's do some like elemental accounting, right? Where do each of these elements of life come from? Well, carbon, nitrogen, oxygen, these are coming from the atmosphere through, you know, photosynthesis, nitrogen fixation, respiration. We get hydrogen and oxygen from water, but basically everything else has to come from minerals in the soil or rocks and stuff like that. Now, does this work on Mars? Do we have the pure elemental composition available to do ecological succession? Well, let's look at the atmosphere. So the atmosphere of Mars has is primarily composed of carbon dioxide, and there's a little bit of nitrogen and argon. So we have carbon, we have nitrogen. These are way, way lower concentrations than we have on Earth. However, they are available. This is something we can work with. This is where the moon does differ. It does not have an atmosphere. So thinking about carbon and nitrogen, this is quite difficult because this is something we probably will have to bring from Earth. Mars also has water, so we can get hydrogen and oxygen from that. And then everything else has to come from regolith. And so does regolith have all these micronutrients for plant growth? Well, it actually does. So if you look at the major plant nutrients here, iron, manganese, magnesium, calcium, sodium, potassium, phosphate, and sulfur, it either has a equivalent amount to what we have on Earth, if not higher. So those pure nutrients required to grow plants exist in quantities that are equal to, if not greater than those in those lava flows I already showed you can support plant growth. So the problem isn't really the quantity of the nutrients, it's the availability of the nutrients. And so on Mars, on Earth, and on the moon, the vast majority of phosphorus, and I'm going to focus on phosphorus because that's what I primarily study, and so I know that the best. It's locked in these minerals called apatite, and so you'll get these apatite as sort of co-minerals with silicates and stuff like that. And these are really insoluble, and plants have a very hard time accessing this. And so what they'll do is they'll form associations with fungi, bacteria, archaea, and other microorganisms to help them dissolve these minerals and increase their uptake. With fungi, we get these fungal hyphae, which kind of break apart these minerals and kind of extend the plant sphere of influence into the soil. And then there's bacteria, which along with the fungi and the plants will produce organic acids and chelating molecules that will dissolve those minerals and help the plants uptake them. So we talked about earlier how it took 500 years to go from pure lava flow to an advanced forest. And while I think that's super fast, that's not fast when we're thinking about how do we create like a livable ecosystem in a biohabitat on Moon or Mars. And so perhaps can we improve this? So that's kind of what I am working on. So basically the strategy I'm taking is collect these endophytes, which are those plant-associated microbes, from places early in ecological succession, which are kind of mirroring Mars, and then isolate them, and then test for their ability to dissolve these minerals and how they interact with plants, fungi, and how they help plants uptake those minerals from the regolith. And so if we look at previous research, we can kind of try to inform ourselves on the feasibility of what I'm trying to do. So there's these three main research papers that came out in the last couple of years, which I think kind of prove that what I'm trying to do is, at least in theory, possible. The first one here is this paper from Paul et al. I think this was 2022. This is the UC Florida, or the UCF, University of Central Florida group. And we saw some people show their results earlier. But basically what they did is they grew plants in real lunar regolith. So this was regolith returned from the Apollo space missions. And they showed that plants could grow in that real regolith, but they showed signs of stress, both abiotic, but also nutrient stress, like phosphate stress, which is interesting because they did apply a nutrient solution to this growth media. And so I'm very curious as to why they experience those phosphate stress and whether or not endophytes can help with that nutrient uptake, because they have so successfully done that in our modern in agricultural systems. This next experiment by Ji et al. Is from this Chinese group which grew tobacco plants in lunar regolith simulant, and they used rhizospheric phosphate solubilizing bacteria. So these are not bacteria that are colonizing the inside of plants, but are kind of in the vicinity of plants. And they showed that these microbes dissolved phosphorus in regolith and were able to help plants grow and uptake phosphorus. However, a limitation of what they did is they used organic phosphorus in their growth media. So I think it's kind of hard to say how much of the phosphorus was coming from the actual substrate versus their additional phosphorus they added, because that measuring phosphorus is quite difficult and complicated. But perhaps this is actually not that bad an idea at all, because, if we're trying to grow plants on the moon or Mars, we're probably going to want to add phosphorus and other organic material to help improve plant growth. The last experiment here is done by Naz et al. This was 2023. This was done by a group at Tufts, and they showed that bacteria could grow in actual Martian regolith. And so this wasn't returned from Mars, obviously, but it was a meteorite that they found, which they found was from Mars originally. And they showed that bacteria could live within it. And since they used water as their growth media, they were able to show that they were able to grow in this media. However, they didn't do any sort of like soluble nutrient assays, so it's hard to tell how much of that was dissolved. So if we really want this to work, and I certainly do, there's a huge amount of room for improvement. Basically, our methods for screening phosphate and also mineral solubilizing bacteria is awful. There's a horrible correlation between bacteria that are screened on plates, like these ones here, also being able to dissolve phosphate minerals in solution, and then trying to connect that to growing plants in a field is even worse. And so we need to improve this method for screening, and we also need to develop high throughput assays so we can test more bacteria. We also need to understand the basic physiology of these bacteria. Although they are ubiquitous and they've probably been important since the beginning of life, we actually know relatively little about their ecology and physiology. We also need to understand how these bacteria play with the other members of that tripartite system, right? We got fungi, plants, and bacteria, and there's very few experiments showing how these interact and which role each of these players play in getting nutrients. Oops. We also need improved regolith simulants. We've heard a little bit about regolith simulants over the last 2 days, but something that they don't really do super well is model toxic components of actual regolith. So for Martian regolith, they don't really model the chromium levels or the perchlorate levels, which are really important if we want to understand how plants can adapt to these environments. We also want to see if we can bioengineer these relationships, but because we know so little about their interactions and physiology, we don't really have candidate genes or mechanisms or stuff like that we could potentially involve. And because it's such a complicated process involving many, many, many genes in multiple organisms, this would probably be quite challenging. So even if you think that my idea of dissolving regolith minerals is crazy or not worthwhile, endophytes are going to help us grow plants off Earth because they can help with organic phosphorus uptake, they can help with other nutrient uptake, abiotic stress tolerance, like Morgan talked about, water use efficiency, and nitrogen fixation, as Dr. Doty talked about, but also pest and disease resistance. So if you're interested in using endophytes for any of your experiments, please email me or my PI, and we'd be happy to do collaborations. So here is my email at the bottom. If you have any questions or want to establish a collaboration or just have any comments at all, you can either ask them now or email me.
Okay, thanks a bunch, Kevin. That was really cool. I think we've still got several minutes for questions, so if the audience has some, please put them in the chat. Oh yeah, here we go. AC asks, how about adding, say, hydroxyapatite as the only phosphorus source in the agar for selecting such bacterial species?
So yes, you can do that, and a lot of people have done that. But the problem is, so if you look at the actual assay that people use to first isolate phosphorus solubilizing bacteria, they form this halo in this material called TCP. It's like a more soluble version of hydroxyapatite. But the problem is this doesn't happen if you use hydroxyapatite generally. I mean, here is an example of it, but it is quite rare. But that problem of linking a plate experiment to a soluble experiment, so like in solution or with plants and fungi, is quite difficult. Actually, just creating a media to grow these bacteria in is quite difficult because all of the other micronutrients you need to grow plant or grow bacteria like iron, manganese, they all react with phosphorus and can really impact the study of the dissolution of these minerals.
Yeah, that is a tough one. Trying to get these extreme lithophiles to grow on a plate is, I mean, that's not what plating is for, right?
There is also a problem that I have noticed where some of these bacteria will grow on plates without any phosphorus at all. You need very, very little phosphorus to grow bacteria. And so if you're not using like noble agar, like it is not going to work because they're just going to dissolve whatever is left over in the agar. So if you're trying to grow any of these bacteria, please email me. I've spent a year trying to figure out how to do this.
Yeah. Yeah, I suppose you can also read the chat now, but I guess, do you know the enzymes that are responsible for the chemical dissolution of the minerals?
In short, no. There's likely hundreds of enzymes involved in hundreds of metabolites that are involved in this process. And so these aren't actually enzymes breaking down the mineral nutrients. They're producing metabolites which go and influence like the Le Chatelier's principles of the minerals, which lead to their dissolution. So I mean, there's enzymes to produce those metabolites, but the enzymes aren't actually the thing doing the dissolution.
Gotcha. I mean, what is if you had infinite money, how would you try to solve this problem?
Tons of omics experiments to try to uncover all the genes.
Sure. Yeah.
But that is the problem, right? Like with something like nitrogen fixation, there's a handful of genes that are involved in it. And so like there's this screaming red flag when you have nitrogen fixation, which is not happening with phosphorus solubilization or any of these other minerals.
Yeah, phosphorus is a tough cookie to crack. It's also really hard to develop sensors for phosphorus. I think Marshall here could talk at length about that. Just a hard element to hold down, huh?
Yes.
Okay. Oh wait, do we have one more question? Okay, yeah. Lawrence has got a good question. He also did some research in Hawaii. He said, have you considered using ohia or other native Hawaiian plants for a project studying ecological succession? But he said
In short, absolutely. I'm trying to get to the picture I had. So right here is a ohia plant. Ohia is very interesting because they colonize these lava flows really quickly. So that is something I'm very interested in doing. So if whoever asked that question could help me with that, please email me.
Yeah, Lawrence, I'm sure, yeah, he can grab your email there. That's great. I know he would love to revisit his Hawaii research days. Did we have another question in the chat, or was that it? I think not. Okay. Well, Kevin, thanks a bunch. This was a great way to wrap up the Doty Lab presentations. I think we all learned a lot about endophytes in the past couple days.
Thank you.
Thanks a lot for your time. So here we are at the last presentation of the Space Ecology Workshop, and it is from my co-organizer, Jane Shvetsov. And she's going to be telling us, the big elephant in the room. How do we actually build a biosphere? How do we start building a biosphere that can support life? So I'm really excited for this talk. Okay, Jane.
Thank you, Patrick.
Yep.
So I enjoyed Kevin's talk, and I was also kind of going, because he said so many of the things that I'm going to say at the beginning. So there's going to be some overlap. So we've been thinking a lot about near-term work or medium-term work, and we've been hearing a lot of great ideas about technologies, ways to grow particular plants, ways to use fungi. I want to zoom out. I want to go really big picture, really long term, and talk about how do we build biospheres. So this is what I'm defining as the goal. A stable, self-sustaining biosphere that supports a long-term human population with minimal inputs, and somewhere this should say long-term. So this is not about Artemis, this is not about things that are happening in the next 5 or 10 years. This is looking, let's say, the rest of the century and after. Like I said, big picture. So I said biosphere. What do I mean by biosphere? For the purposes of this talk, I'm going to define it as a mostly materially closed system in which most flows of the elements that are necessary for life are driven by organisms. So those mostly are important because even the Earth is not completely materially closed, almost, not quite. And some flows like are not driven by life. I'll mention that in a minute. So it can be as big as the Earth. It can be as small as this ecosphere I used to have on my desk. So we're really thinking about flows. Now, one thing that I really want to mention is that biospheres, or the biosphere, we only know one natural one, include non-living components. People think, oh, biology means only biology. But if you look at how the Earth works, that's not how the Earth works. That's why when we talk about nutrient cycles on Earth, the technical term for them is biogeochemical cycles. Because yes, life is important, but then the elements are in the air, they're in rocks, there are abiotic flows that take place. And in space, we're gonna have biotechnochemical cycles. So the biogeochemical. So let's just think about it for a minute. Volcanoes, they melt the rocks, they release gases into the atmosphere. What's the technological equivalent of a volcano? How about this? A very high temperature, I think, oxygen, I think it's called, incinerator. That is gonna take pretty much any material you can put in it and degrade it into constituent elements. I would call something like this the technological equivalent of a volcano. So I would argue that incorporating abiotic elements is not cheating, it's doing exactly what the Earth actually does. Okay, so with that, I'm gonna quickly go through some principles. I think for this audience, we don't need to spend a lot of time on this. But just abstracting the fundamental structure of ecosystems, we're gonna have producers. They're the ones that capture energy and make it available to the rest of the system. We're gonna have consumers that are feeding on the producers. And we're gonna have detritus and decomposers that break down the detritus and release the nutrients, release the elements that are in it. Technically, you could just have these two, but we're gonna have at least one consumer. That's us. We might choose to have others, like Bryce was saying a few talks ago. Having other consumers can speed up processes that would otherwise take a long time and therefore make your system more efficient. Animals are enzymes. Animals are catalysts. They don't change what fundamentally happens, but they can speed it up. And you can see why I was kind of chuckling during the start of the last talk, because the thing that I think we really need to keep in mind is chance. What are organisms made of? So these 6 elements, the carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And as a summary, we do get low to moderate amounts of these things on Mars. That's probably the best choice in terms of element abundance. You get some of these in some asteroids. I've done work on using carbon from organic, from carbonaceous asteroids and breaking down the toxic compounds into something that's usable for soil, for example. And all of these things are almost absent on the moon, especially these first 4. So this is going to be important. And the framework that I'm going to present is 100% a successional framework. So I'm going to make a few assumptions. The context here is a lunar or free space habitat. Free space means constructed and you just put it somewhere in space that you want it to be. Mars is a little different. And also that we are in a place that is relatively easy to resupply. So again, I think a strategy for Mars, which has more elements and is harder to resupply, would be somewhat different, although it might use some of the same ideas. I'm assuming that this habitat, this settlement, is growing over time, and that there is an ample energy supply, at least that we're not severely energy limited. So when I was making this table, I was like, wait a minute. I read this in grad school. Thank you, Eugene Odom. If you know Odom's strategy of ecosystem development paper, this is ridiculously similar. But just taking a very high level view, in the early stages of this biosphere, of this settlement, biomass is pretty low. You've got the humans, maybe you have some salad crops, maybe you have some microbes that are helping degrade some wastes, but there's not a lot of biomass. and flows are primarily machine driven, so physical chemical largely. And you're relying on active controls to stabilize your system. And there's not a lot of cycling. You probably have some, but not a lot. And the idea is that in the long run, you've got much larger biomass stocks. The flows are now primarily biologically driven, with the abiotic ones taking on more of a supporting role. Also, you've got your nutrient pools and you got your buffers that are big enough so that you don't have to be super precise about control, because you've got enough buffer size to deal with a lot of perturbations passively. Again, most likely you will always need some control, but less. And there's a high degree of cycling of the various elements. Okay, so how do we get from here to here? I propose a successional sequence that is element driven. So I think phase one is the biggest departure from typical ideas, because I think all of you, if you talk about space biology to people who don't do this stuff, you're gonna think, they immediately think about plants. And even we think a lot about plants. Plants are important. But considering how limited we are by elements, I think phase one is not about plants at all yet. Phase one is primarily going to be about elements, accumulating elements. And where are those elements going to come from? Well, the easiest place to get elements is human outputs. You store feces, you store concentrated urine. You maybe could start going to plants to capture CO2. I could even see using abiotic materials to capture CO2 or to capture the carbon. The carbon is what you need, and accumulate it for later use. So you might have a salad machine or something like that at this stage. And that's cool, that's great, but that's actually not what's driving your biosphere development. What's driving your biosphere development is this. Typical outputs, elemental, for one human. About 0.3 kilograms of carbon per day, about a tenth of that of nitrogen, mostly in urine. Hydrogen and oxygen are primarily in the form of water, also in CO2. So I broke this down by element, but a lot of these are in CO2 and in water. So this is what you get for free. Of course, you can add stuff, you can strip up fertilizer, you can strip up elements, but this is what you are naturally inherently getting. And these outputs are either a solution or a problem. If you don't make them a solution, then they're a problem. So you might as well do something useful with them. And at this stage, you are collecting them primarily in order to start establishing your biosphere. Then, and these are not discrete phases, there's definitely going to be a smooth transition from one into the other. One phase might start while the previous one is still going on, but conceptually it's useful to have these phases. So then your first goal after you've accumulated some elements is to establish a carbon cycle. So you've got some plant substrate, you've got enough CO2 to be growing a substantial amount of food, maybe other vegetation. And you are recycling not only the exhaled CO2, that's fairly easy, but also the plant parts that we're not using get composted, and hopefully also human feces, which are primarily carbon, actually. And you gotta do composting for two reasons. One is to establish nutrient cycling. The other one is to use up the extra oxygen. Because if you, as Bryce mentioned, a frequently unappreciated point is that if you are growing most of your own food, you are way overproducing oxygen. If you're not using it up, you're gonna have a problem. So those are your two components of the carbon cycle. The respiration photosynthesis one, and the composting of various organic materials. And at this point, you're using probably a reasonable amount of inorganic fertilizer. That's some mass, but it's not an enormous amount of mass. Now, at stage 3, you can establish the nitrogen and phosphorus cycles. And for humans, if humans are your major consumer, that means that urine processing becomes very important because that's where most of NNP comes out. And some of this was already happening in the previous stage with composting, but urine is a whole different thing. It's much more rich in these nutrients. You gotta watch out for that. It also contains sodium in fairly large amounts that you need to separate out and then either store it or incorporate it in the human diet. The thing about sodium is not that it's difficult to resupply. We don't need enormous amounts of it. It's that if you don't separate it out, it's toxic to plants. And if you do separate it out, then you have now mounds of sodium that you gotta do something with. So might as well find a way to cycle it back. I have a proposal in the review right now about a way to do that. But at this point, you're really focusing on establishing the nitrogen and phosphorus cycles in your biosphere. And yes, of course, that can include inputs from your environment, like Kevin was discussing. So now you're cycling all your key elements because hydrogen and oxygen cycle along with these guys. And having established those cycles, now you're focusing on growth. You're focusing on increasing your buffer sizes. So the plant population, the human population, the tidal biomass all grow. And so do the abiotic stocks relative to the population. So you slow things down and you increase the buffers, and that improves the system's ability to handle perturbations. Okay, so that's kind of the successional framework. Now, there's one aspect of this that I really want to discuss, and that's element scarcity and element ratios. So I'm an ecologist, and in ecology, we think a lot about C to N to P ratios, especially in plants. Basically, if you have more nitrogen per unit carbon and more phosphorus per unit carbon, you're gonna have faster decomposition, and often also these are associated with faster growth. So why does that matter? Because there is not a lot of nitrogen in space. Nitrogen is probably going to be your biggest limiting factor in terms of large-scale human settlements off Earth. How do you deal with that? Well, I think we can take a cue from tropical rainforests. These are some of the most productive ecosystems on Earth, but the soil is notoriously poor. In fact, it's primarily phosphorus limited in a lot of places. If you cut down the tropical rainforest and you try to do agriculture there, that soil is going to last you 3 or 4 years before it's too depleted to grow much of anything. So how do tropical rainforests work? How's high productivity on very poor soil possible? Answer: rapid cycling. The nutrients are not in the soil, they're in the plants. They're in the biota. So you've got organic matter that as soon as it hits the ground, it starts to degrade. A leaf falls or a tree falls, and boom, all the insects, all the fungi, all the bacteria are on it like that. And the plants, with the help of microbes, are able to rapidly take up those nutrients that are released. So the total amount of nutrients is not large, but the cycling is fast. And that rapid cycling is what enables high productivity without a large nutrient pool. I think this strategy, this analogous strategy for nitrogen, can help alleviate nutrient limitation. And I think this is going to be what unlocks large-scale settlements. I did some very simple mathematical modeling of this. And on a relative scale, you can see this is the shape of the distribution. This is not even a simulation, this is analytical. That as you increase decomposition rate, the amount of plant biomass that you're able to have rapidly increases and then levels off. So don't worry about the numbers, but look at the shape of the relationship. That's what's going on. So we want to be at least about here. And establishing rapid cycles to get around these inevitable limitations. Okay, one more topic that's very much on my mind, and that's stability. Now, there's a bunch of different things that people mean by stability. I think there's a paper that I think catalogs something like 30 ecological concepts of stability. But here's the big two. Resistance is being unchanged by perturbation. something happens and you just don't react very much. Resilience, which for some reason is the one that's getting all the love at the moment, is you have a reaction, might be large, but you quickly return to normal. So how does the closure of an ecosystem, of a biosphere, affect resistance and resilience? Well, there's some interesting work that I found in the literature that has nothing at all to do with space. These were just people doing normal ecology in the 80s and early 90s. And they found that resilience decreases with system closure, but resistance increases. The names to look up are Laro and DeAngelis. We'll put it in the chat later. And furthermore, there's a mathematical relationship between the return rate after perturbation and inputs and biomass size, return amount. And it's just that the return rate is proportional to the ratio of inputs to nutrient cycles, so to nutrient amounts. So if you lose some elements, which is the type of perturbation we're talking about, then actually a lower input helps you return faster and slows down, and you become more resistant to these perturbations. So there's actually a mathematical relationship between the return rate and sorry, I didn't make that very early for this. Now, this is about perturbations to the various stocks, which is what there's a lot of mathematical theory about. You know how to do this. There's much less known about changes in rates, even though these kind of perturbations are arguably the more realistic ones. So this is one place where basic work is needed. So a few future directions. One, we need to develop soil or plant growth media, plant growth methods that support nutrient cycle closure. I like soil partly because you can do composting, but maybe there's other ways. But supporting nutrient cycle closure, that's non-negotiable. We need nutrient recovery methods, and we need fundamental work, both empirical and theoretical, on stability and control. Control of ecosystems is not something most ecologists think about. I know I am one. So, but for long-term space settlement, this is the kind of work that needs to be happening. And okay, that is the very big picture.
Thanks for that big picture look, Jane. That's an excellent way to cap this workshop. Your presentation has already spawned some talks in the webinar chat about how we're going to get these elements for our biosphere, which could be a whole nother conference itself. Do you guys have actual questions for Jane, or are we just going to keep debating the where we're getting our nitrogen from? Actually, Jane, this is actually pretty relevant to you as well, because I remember you had a NIAC for the fungus making soil out of asteroids, which is
yes.
Yeah. So given your I guess, experience in finding elements for these space habitats. Where do you think we should get all of these rare elements from?
Most of them you can get somewhere in the inner solar system. So asteroids are often good sources. Beyond Mars, you have at least moderate amount of elements,
Hmm.
with the exception of nitrogen. There's no good source of nitrogen in the inner solar system other than shipping it from Earth, which
Hmm.
you can do to an extent. Of course, you have to do it to an extent. But once you're thinking long term, you cannot be shipping enormous amounts of nitrogen. So this is not something I think would limit you, say, in the next 20 years, but it will be limiting you in the next 200 years.
I see.
That's why I was focusing so much on cycling.
Yeah, yeah, that makes sense. But I guess if you want to increase your buffer capacity for big populations, that's when we'll have to start thinking about it. But maybe at the 200-year scenario, we'll be in the outer solar system and we can just scoop it out of the atmosphere of Titan or something.
Yeah, the gas giants have pretty big gravity wells.
Well, yeah, that's true. Titan's smaller than Saturn's, though.
Yeah, but it's a moon of Saturn. You're still in Saturn's gravity well. But maybe. So there are sources, but none of them are great.
Hmm.
So, like AC said, it's not that you do get some inputs. It's not that your input is zero. And by the way, the main limiting factor in most tropical forests is thought to be phosphorus. I think that's still the case. But in both cases, um, it's not that your input is zero. It's that it's quite small relative to what you need.
Sure. Do you think in this conversation we didn't talk too much about all of the micronutrients that we're going to need? Do you think that those are available in sufficient supply wherever we might be?
I haven't heard of them being like too hard to get, because the nice thing about micronutrients is that they're micro, you don't need enormous amounts of them. But they do present a different challenge, which
Mm,
is recycling. Because even micro is going to be a problem when it builds up in one place. Like the one that I'm really thinking about these days is sodium.
yeah,
Um, it's not that you couldn't resupply your settlement with sodium. I'm sure you could. It's that if you don't deal with it, it's a pollutant. A pollutant is any substance in a place or in an amount that you don't want it to be.
right. And I guess now that we're talking about micronutrients, I don't even know, are there biological cycles for some of the really, really tiny concentrations? I don't know, like selenium or something? Or is that always found in mineral form and not recycled?
I mean, nature does recycle everything. It's inevitable, because if something is dead and then the nutrients are released, then you're gonna release the micronutrient as well. Now, to what extent they're limiting, there's some interesting work that suggests that sodium might limit animal abundances because plants take it up so little.
Huh.
Potassium would rarely be limiting, that's kind of everywhere. Um, Iodine is an interesting one, because we know that in certain places, humans will often be deficient in iodine unless it's supplemented. That's why salt is iodized. And
Mhm.
that causes thyroid problems, and if deficiency is severe, it causes neurological problems like with brain development. So we definitely are aware of cases where on Earth where micronutrients can be deficient.
Sure. Okay, that makes sense. we have any questions for Jane?
Yeah, but I think that's precisely what I was talking about. The there's not a lot of stored elements, but they get released quickly and they get taken up quickly. And I'm sure there's all kinds of microbes driving those processes.
Okay, well, I guess if there's nothing else, Jane, thanks so much for this talk and thanks for your help organizing this workshop, of course. It was a pleasure. I think we're ready to move into the final stage of this agenda of this program, which is the closing remarks. Um, I mean, I guess these are the closing remarks right here. I don't have too much to say other than, well, thank you to everybody who made this event possible. All of the speakers are featured speakers as well. I'm really grateful to you all for volunteering your time and your expertise to share it all with us. Thanks, of course, to all of the attendees so that we had people to share with, and for providing such excellent questions and for joining us in this. I guess talking about the future, of course, we're going to do another one next year. It's kind of a given at this point. We have discussed pretty extensively. Actually, we spend like half the year deciding if we want to change parts of this workshop, and then we kind of just end up doing the same thing. We talked about like starting a journal, for example, and we went a little bit down that path, and then it didn't really work out, because Sherry, who was one of our organizers in the past, she had to step back. Actually, she got hired at a real science journal, and so she didn't want to have the conflict of interest of running a space ecology journal, which is pretty reasonable. Yeah, so I guess you guys can expect more of the same next year. If anyone is interested in helping out, they're welcome to email me. But to be honest, is not that much to do. You just host a Zoom webinar. Maybe we could have help like spreading the word. That is always nice. I think this year we had slightly less participation than last year, and that's probably my fault because I did not spend so much time promoting it. I was traveling a lot this summer. Yeah, but I think that's everything. So again, thanks everybody. All of the presentations were fantastic. I learned a lot. I had so much fun.
Excellent, excellent presentations. I think the quality has actually gone up.
Yeah, I think so too. And this was also the most presentations we ever had. We had 30 this year, which is crazy that we didn't have any gaps in the presentations either, despite all of the injuries and the hurricanes. So thanks we did have a couple people sub in, which was really clutch. Thanks.
That was amazing.
Yeah. All right, so let's get out of class 15 minutes early. Thanks again, everybody, and I will send all of this. I'll send a follow-up email with the links to the recordings and the program and the chat log downloads and everything, or you could just come back to the spaceecologyworkshop.com to find it later. So until next time, everyone, I'll talk to you later.